Transcription is the process of transferring genetic information from DNA to RNA. Transcription uses an enzyme called RNA polymerase to unwind the DNA. The enzyme builds the RNA and rewinds the DNA. The strand of DNA that is being copied is called the antisense, and the strand that is disregarded, for this example, not copied, is called the sense strand. Next, the RNA elongates through the synthesis process, and when finished, the RNA strand peels away from the DNA template, allowing the two separated DNA strands, sense and antisense, to come back together.
The next step is RNA splicing. Genes of plants and animals include internal non-coding spaces called introns, or intervening space sequences. In our model, these are colored black. The parts of a gene that express amino acids are called exons. Before RNA leaves the nucleus, the introns are taken off and the exons are joined together to make an mRNA molecule with a continuous coding sequence. This cutting and pasting process is called RNA splicing. Next, the mRNA exits the nucleus and moves along to the ribosome, where it will begin a process called translation. mRNA is read in three-letter words called codons. Each codon represents an amino acid. Transfer RNA molecules, or tRNAs, have three-letter words, and they are called anticodons. They match with codons using the base pairing rules. tRNAs wait in the cytoplasm until they see that their set of anticodons can complete the match of the codons on the mRNA. The tRNA moves into the A site. Then the tRNA moves to the P site as another tRNA moves into the A site. This is the start of the elongation process. The first tRNA moves out through the E site, and while this is happening, passes its amino acids to the tRNA now in the P spot. This process repeats itself until a chain of amino acids is formed and therefore creating a protein. The last step of the translation process occurs when the mRNA reaches a stop codon. The amino acid chain then releases from the tRNA, and the small and large part of the ribosome tear apart to let the protein go out into the rest of the cell.